from now. So I came across this idea of using inhibition of interleukin 11 to extend lifespan in mice, and a university over in Israel discovered this. And I'm going to talk mostly about how this inhibition works. And then I'm going to talk about more general conclusions as you can get from looking at this. So interleukin 11 binds with interleukin 11 RA, which binds to GP 130. And these are all different proteins. And these arrows mean that when it binds, it activates it. So as you come down this, you can see, you know, it has this complex interaction with all these other proteins. And this mTOR is actually a protein complex, and we'll get to that in a while. But these other things are just proteins. So once it gets down here, the STAT3 stands for signal transducer and activation transcription 3. And it has a role in a lot of cellular processes like growth, division, and cell apoptosis, which is programmed cell death, by activating all these other interleukins and CCL2. And that's kind of the end of the story for this leg of the interactions. So ERK stands for extracellular signal regulated kinase. And this is another protein, just like all of these other things. And this kinase inhibits LKB1 and promotes P90RSK. This LBK1 is kind of a critical protein. So if you inhibit this, and because this LBK1 stimulates AMPK,
recording in progress.
The AMPK is reduced if IL-11 is increased. And AMPK inhibits mTOR. Well, I'm getting kind of on here.
Let me.
Okay.
See if I can fix it.
Yeah. Greg is working on
there.
Okay, good. So if mTOR is inhibited, that reduces the production of P70, S6K, and S6RP, which has interesting consequences for cellular growth. So LBK1 stands for liver kinase B1, and it's an energy sensor that activates AMPK, which is a key source of energy for the cell. And by activating it, it increases cellular energy levels. So by inhibiting this, IL-11 reduces stimulation of AMPK. AMPK stands for adenosine monophosphate activated protein kinase. And I actually learned about quite a bit about this cell when I took my cellular metabolism class last spring. So AMPK acts as a detector that senses low levels of adenosine triphosphate, which is the main cellular energy source. And if ATP is low, then AMPK stimulates metabolic pathways like fatty acid oxidation and glucose transport, and inhibits ATP consuming processes. So if IL-11 is high, it will inhibit LBK1 LKB1, which also inhibits AMPK, thus reducing the amount of energy that a cell has. So this red line means that with a bar, means that the mTOR is inhibited by high AMPK. And inhibition of mTOR is a key way of extending lifespan. And mTOR can also be inhibited with rapamycin, which is why mTOR stands for mechanistic or mammalian, depending on who you talk to, target of rapamycin. B70S6K is increased by mTOR increase, which increases S6RP, which is a ribosomal protein activator. And this activation stimulates the production of proteins. So I want to take this and put it in a larger cellular context. So we've been talking about some of these things here: LBK1, AMPK, mTOR, P70S6K. And you can see how these kind of fit into this network that all centered around mTORC1. And this mTORC1 takes signals from outside the cell, and it gets processed through some of these other proteins and changes how it reacts based on signals from outside the cell. And it also takes signals from inside the cell and local energy status, which comes from the AMPK, and communications from mTORC2, which is another key regulator. And these two things talk back and forth to each other quite a lot. And what all this does is it allows mTORC-C1 to put out control signals to the cell. And one of them is this P70S6K that we talked about a little bit, and I put stars by the ones that we talked about earlier in this. And that stimulates cell growth. And it can also stimulate autophagy, where the cell commits suicide. And this suicide is committed by detecting errors in the cell. And one of the errors that it can detect is beginnings of cancer. And of course, you want to get rid of any cell that is starting to get cancer so you don't wind up with cancer. And mTORC-C1 can also directly stimulate autophagy or cell suicide, and it is a key thing for proliferation or duplicating the cell, and it gets involved in fat or lipid synthesis and ribosome biogenesis, which is one of the things that this P70S6K does. And what ribosomes do is they are what transcribe the DNA and get the programming for making your proteins. So if there's lots of ribosomes around, you'll get more DNA synthesized, and so you get more proteins. And that's one of the key things you need in order to get to growing new cells. And of course, if you're doing that, you need to do RNA splicings to make sure that you get the right section of gene that's being converted into a program. And of course, you increase the transcription of all this stuff. Lipogenesis is also affected, and this list is all over here on the right-hand side. And adipogenesis, which is making new fat cells. So if this was a fat cell, it would encourage the cell growth and storage of fats. Mitochondrial metabolism, which is your primary energy source for the cells. Angiogenesis, and I thought this was a very interesting one because that's where your blood system grows new arteries and veins. And it's kind of key as you get older to maintain this, because if you don't, they eventually, I don't know if wear out is the right term, but you start having reduced blood flow. And that is a pretty big problem if you're trying to maintain your blood supply to all your organs and stuff. And this also controls lipid metabolism. So you can see that this mTOR C1 is a key thing. And if you inhibit it with, say, rapamycin, which I've talked about quite a lot during these lectures, it reduces this mTOR C1. And because of that, it slows down a lot of these things in this list. And a lot of these things sound like they're pretty good, but the end result is growth, and not necessarily good growth either. So as you get older, if you slow this mTOR C1 down, we're using rapamycin or the IL-11 that we started this discussion with, then you will tend to live longer. And that is probably what's going on with these mice that are living longer. So this kind of opens up something that I've been thinking about for quite a few years. If you look at these patterns of protein interactions, you can see they're primarily command and control networks that keep everything from the local network inside the cell to the whole individual functioning. And each one of these networks, and you can see that we started out talking about this little network here that controls these four things, to talking about this entire network. But in actuality, each one of these proteins is connected to a network, and you only see some of these networks because this AMPK1 probably connects to a lot, dozens at least, and possibly as many as hundreds of different networks. And you can see how complicated this would get over large, or if you connected all the proteins in a cell through their networks. And you can see that these networks can be seen as programming, somewhat similar to programming a computer. And these networks become very, very complex as you expand the part of the network that you're looking at. And if you look at the entire cell, we understand so little of these networks that it's we can't even predict very well, other than pieces where we picked apart the exact mechanisms of what's going on, the overall result of the network in an entire cell. And of course, you can even expand that network out to include how the cells network with other cells. And so the programming involved in this is extremely complex. And my hope is that our artificial intelligence will be able to crack it and tell us what happens as we age, so we know what to fix. And it's my feeling that this program that forms the basis of life is perhaps deliberately flawed, and that results in aging. And deliberately is perhaps not the right word, but my feeling is that this flawed aspect may be because of the need to drive evolution over the long term. And that's the end of my slideshow. So let me stop the recording.